Yeah. So I thank my good friend Dr. Prash and Professor Ganesh Chaudhary and the whole organizers for making such a spectacular arrangement. Uh, when the program came out, I must digress and say that I was a little nervous and a very nervous man today because I found that I had been pitchforked into an evolutionary genetics session. But then in the morning, Dr. Raghav was leading a discussion that while you are in Kashi, you have to be spiritual. You have to look within yourself. So let me take this opportunity to change gears. We have been looking at populations as individuals. Let me now start getting you thinking in Kashi as populations of cells, that is population within our body. And what else? But the human body is a glorious example of this internal or the internal universe. So we have about 30 odd million trillion cells which are human in our body, as we all know, and 30 odd trillion odd cells which are non-human. No wonder in my childhood days I was always told by my seniors that I have a strong non-human origin, but maybe from different other components. But all these cells have genomes, and of course the same genome. And these cells also, the genome gets expressed, and the transcriptome, the gene expression machinery that expresses about 20,000 odd genes from each of these cells, create that polyphony, as you would like to call it, that combination of music that actually determines our phenotype, keeps us healthy, makes us fall ill, eat certain kinds of food, and so forth. So what we try to also understand that these cells are the individual executors of genotype to phenotype through the transcription. But when we look at this bulk kind of data, we generally take a whole bunch of cells. We are not able, and the cells have a lot of variations, just like in an orchestra, you have about 100 of the large orchestras about 100 odd equipment, but you all you hear is that polyphony. You don't get to realize how each of these instruments actually contribute. So and to understand this polyphony of these cells and how each cells talk with each other, express genes in a different way, a tumor is a very exciting example. Our ecosystem consists of a diverse kind of cells. The malignant cells themselves have, now we get to understand, have different cells states, what we call as. And then we have also different kind of non-malignant cells, which are very exciting. They have immune cells, we have stroma, and then many of these cells talk to each other, and the way they express each of these cells gives us an opportunity to understand how the tumor grows, or when it will metastasize. And we now understand that even drug response or the prognosis of the tumor is determinant of this expression. So many of us took this challenge of using the newer technologies of single cell transcriptomics to understand how the tumor cells actually create this environment which results in many of these features. Now, when we talk of tumors in India, we talk of oral tumors, of course, because still the largest tumor cause of cancer of men in India, primarily driven by chewing of tobacco, happens in the gingiva collapses, and that's how it's predominantly different from the West. And we started working on single cell transcriptomics. We started to use oral tumors because we had been generating a lot of genomic and transcriptomic data on this in the International Cancer Genome Consortium. We also, when we started this work, almost serendipitously, we found that there are a certain set of oral tumors which appear on a background of a condition known as oral submucous fibrosis. Now, this oral submucous fibrosis, or OSMF, is very common in India, and it's caused primarily by chewing of tobacco in combination with areca nut, and it's very painful. And we find that some of these people also go on and develop oral tumors. We started to find differences. The community is divided between whether oral tumors developing in a OSMF background has a better prognosis or a worse prognosis, because the clinicians are completely divided. Some say they are better, some say they are worse. So we have some data here, and the jury is out, and at the end of the day, I will go on to ask you to make your own conclusions on what is the difference between these kind of tumors. So that brings me to the talk that we'll be looking into the cellular polyphony of the tumor ecosystem of oral cancers and OSMF. Uh, I, there are a lot of funders and everybody, very established people here whom I don't need to introduce, but I will just point out that this whole work is spearheaded and fueled by young people. The young postdocs and the PhD students in the lab who over the last 5 years have been working on this and are still continuing to work on this. And of course, clinicians who from a dental college in Kolkata who have been actually came up and started partnering with us in this work. So what we did was, after our initial studies, we actually worked on these oral tumors. We looked at treatment-naive patients who came to the dental hospital and diagnosed with oral cancer there, and we actually did single-cell transcriptomics of the tumors from about 12 OSCC who developed oral tumors without any concomitant other kind of conditions, and about 15 OSMF associated OSCC. So they are OSMF patients where the oral tumors have developed. So the work was primarily done initially by my young postdoc, Dr. Sillery, who is now in University of Chicago, Sumitago, who just 2 days back submitted his thesis, and now being taken over completely by my senior PhD student, Arindam. So what we found was that we had about 27 samples, and we found various kinds of cell types. So here's the UMAP. Each dot represents a cell, and the two dots are close together if they are similar across 20,000 genes expression or far apart. And then we use standard, well-known genes to actually label these clusters as what kind of cell types they have. So what we also did was did a deeper analysis. So we had nearly about 100,000 cells from this analysis. So which allowed us to do a deeper analysis and identify something known as cell states. So cell states, unlike cell types, which are like you and me or anybody else, the cell states can be different. Right now I am nervous. Next moment when I have food, I'll be very happy. So these are different kinds of cell states. So we found that each of these cell states actually had very different kind of cell state. We identified about 9 distinct cell types and 43 distinct cell states from these oral tumors. And what I would like to talk to you about is only, there are many things that are very exciting, but only 2 things. One is some are observations on the malignant cells and certain subset of immune cells. So when we looked into these malignant cells, we actually subclustered them. We can do that. And we found that within the malignant cells, there are 4 distinct cell states based on the gene expression differences within each of these cells. And these 4 distinct cell states come up when you recluster them as 4 distinct clusters. We actually found that actually they belong to 2 patient subgroups, these 4 clusters. And when we looked at the differential abundance of these cell states, we found that in this excess, there is OSMF associated with OSMF associated with OSCC here and only OSCC here. We can see that there are 2 different clusters, like cluster 2 and 4. They are almost exclusively associated, found in the OSMF associated OSCC, whereas clusters 1 and 3 actually are present in both. That means within them there are 2 distinct again cell states. One is associated with OSMF and the other is not non-OSMF. We looked into the differential gene expression and found that clusters 2 and 4, which are more associated in the OSMF associated OSCC, they actually are associated with upregulated cell migration and also from immune signaling and cell death. So we seem to see, whereas clusters 1 and 3 actually have related to metabolism, stress adaptation, and maintenance of tissue structure. So it seems that there are more immune activity that are associated with OSCC OSMF background OSCC compared to just only OSCC. So this was one observation. The second was, we then started to look at what are the kind of gene expression signatures each of these cell states represent. So what we found was very strange. These are all malignant cells, right? And these malignant cells, these are epithelial cells. But we found that the cluster 1 actually expresses genes which are related to immunoglobulins. So they are expressing immunoglobulin genes. So we call them as IGKC. Cluster 2 primarily had expressions which relates to a transition state. So in cancer, we know epithelial to mesenchymal transition, EMT. But what we are seeing, and I'm not showing detailed data of that, is what we are seeing something known as partial EMT. So the cells are transitioning from epithelial to mesenchymal, and we are catching them in that point of time. And then we also found two clusters which are expressing fetal genes. So they are going back and starting to express fetal genes, and we know that the partial EMT again is known in cancer biology to be a a hallmark of poor prognosis. The fetal gene expression of malignant cells are known to be associated with immunization or immune suppression. So we see that there are different cell states, and these are very important in terms of the way they affect the tumor. And these are all malignant cells. So what we did was, again, evolutionary session, so I have to show you some evolution, but this is not in real time, but this is in pseudo time. So we had, it's an assumption that if we have large number of cells, then we'll find cells in various states of transition. So doing that, we can actually draw a trajectory, and we found that all these four clusters actually occupied the four corners of a trajectory of a pseudo time plot. Where PMT and IGKC are on this side, and these two branches are essentially contributed by the fetal gene expression. In one case here, there is the cells here express both partial EMT and fetal gene expression signatures, whereas in the other there is only fetal expression. So branch 1 is more associated with immunomodulatory pathways, whereas branch 4 here is associated more with cellular metabolism, which is more aggressive. So branch 1 is essentially with the OSMF associated with CC. Again, I mean, take your own call. We are still not able to figure out. To us, it seems, and I will show you some more data, that OSMF associated with CC might have a better immune response. Whether it results in a better prognosis, we do not know. So we also, from this data, we identified two signatures, malignant cell gene expression signatures, and we looked into the TCGA data where they had large number of OSCC patients with a follow-up. We found that in both these signatures were actually associated with poor survival of the patient, overall survival. So we are right now looking into our own patient data set in ICGC and trying to see if we see the same trend. Unfortunately, our data does not have as good follow-up as the TCGA, but we are still trying to have enough numbers to do this kind of analysis. So this was malignant cells. The second one, I will shift gears now to immune cells, because oral tumors are known as hot tumors. You have a lot of immune cell infiltration. So no wonder that when we looked into this data set, we found that there are a large number of T cells. Really large number of T cells. And what we did was we looked into these T cells, and we did subcluster them to understand what are the different states. And we found that we could find a large number of states, like we have Tregs, we have various other kind of natural killer cells, and then we have CD8 cells, so all kinds of cells are there. What we found interesting was this particular cluster, which we call them as double negative T cells because they do not have expression of CD4, they do not have expression of CD8. So, and that's why we call them as double negative T cells, and they of course express CD3. And then what we did was we subclustered them again and found that there are three clusters. One where the so we found that there is an expression of a particular gene that some of these cells have a very high, which is called PLCG2, phospholipase C gamma 2. And we found that there are three groups of these double negative T cells. One that is PLCG2 low, PLCG2 high, and cells where there is an intermediate level of expression of PLCG2. We then also looked into the other cell types, and what we found was that the OSMF associated OSCC all had cells with at least T cells which are like associated with CD8, association with their natural killer T cells and so forth, which suggests that there is more immune activity going on in these tumors. Might again adding to our story that there is more immune activity going on in OSMF associated OSCC. What we also found from this was that both the PLCG2 high and PLCG2 low double negative T cells were almost exclusively present in OSCC samples, but not in OSMF associated OSCC. This was striking. And then what we did was we looked into these, and so this shows the differences in the PLCG2 expression. And what we did was we looked into the trajectory. So we found that there is, so cells, difficult to give direction to these plots. What we assume is that PLCG2 high cells are going through a mid, and then PLCG2 low cells are going through a PLCG2 high expression. And in parallel with that, we see a difference in calcium signaling response. So it seems that with the increase of PLCG2 expression, we have an upregulation of calcium signaling response. So question is, does the upregulated calcium signaling result in transition from the PLCG2 low to PLCG2 high? We then looked at cell-cell communication. Generally, we look at expression of the ligands in one cell and expression of the receptor genes on another cell, and we say that two cells are talking to each other through a particular receptor ligand kind of interaction. So if this is a typical fibroblast in our data set, where you can see there's a lot of interaction. Fibroblasts are talking to multiple cells. There's a lot of, it's like if you just in between sessions, this room will have that kind of magnitude of interactions. We looked into double negative T cells, and we find the room probably as it is now if I stop talking. So very little interactions happening. Particularly the double negative T cells are supposed, so here you can see that there is a lot of communication with malignant cells. Malignant cells talk a lot. But here we are finding that the double negative T cells hardly has any whispers of interactions with the malignant cells. So shows that the double negative T cells are not in a talking mode. So then we looked into again something interesting. We found that with the upregulation of PLCG2, we found there's an increase of expression of genes known as BTG1 and BTG2. Now these genes are known for known associated markers of T cell quiescence. So what we feel is that based on the various gene expression data, that there is, this is a hypothesis. Now the challenge for us is to prove it in future, that there is a tissue hypoxia that results in ER stress, which ultimately results in release of calcium, and that upregulates. Now what happens is the T cells are indeed infiltrating the tumor, but when they infiltrate, maybe the CD8 plus cells are losing their CD8 expression. And in presence of high calcium, there is an increase in PLCG2 expression, and increase in PLCG2 expression is resulting in increase in, there is a concomitant increase in quiescence. I'm almost done. And that is the cause. So what we are thinking is that all this is leading to quiescence of cells. Too many T cells coming in, but most of them are getting into a quiescent state and being double negative. So really it's not helping the tumor. And we know that oral cancer does not have a good prognosis in India. So we are just not to ensure that these are not figments of analysis, we also started to do some basic experiments where we actually isolated the double negative. We could show that the double negative T cells are present in the tumors. There are expression of PLCG2 expressing double negative T cells, and what we found was we were trying to isolate these double negative T cells, and when we exposed to calcium, we are seeing some increase in PLCG2 expression also. So these are very basic. We are just trying to prepare ourselves to prove our hypothesis, but this is where it stands as of now. And we also found we only developed two gene expression signatures of double negative T cells, PLCG high and low both. In the TCGA data, they are associated with poor survival. So this is what we found: diversity of malignant cells, and there are different malignant cell states which are associated with poor prognosis, and PMT oncovital reprogramming and IGKC expressions are unique signatures of these cell states. We also found that the DNT cells are abundant in OSCC, but not so much in OSMF associated with SCC, and PLCG2 expression might have implication for T cell presence, and hence that can be a potential target. So I'll stop here. We understand our whole lab now is kind of has a strong motivation that this kind of gene expression value is various systems are related to human health and disease. We are working on oral tumors, some of the data I showed you. We are also working on placenta in terms of understanding preterm birth, and we are also working on peripheral blood immune cells to define the healthy state, which is important to then understand disease. This is part of Asian Immune Diversity Atlas, which is funded by CZI. And the question that the lab answer asks is new questions that we have not been able to see so far. These are new data that's coming up. Where do we go from here? Thank you very much. Happy to take any questions.
So, thanks, Arindam. It was very good question. Okay, please.
Even sir, since you have touched upon the important concept of immunity, like a strategic question, the 2023 Nobel is went to Patrick Clark from Blue Eastman for having essentially developed a drug against drug for immortality. How do we gauge its market value? The work that they're publishing journal immunity.
Okay. So let me give a disclaimer. I'm not an immunologist. And the second disclaimer, I'm very poor in history, whether it is science or anything else. So I generally forget Nobel laureates' names, no disrespect to them, but I cannot track these things down. But what is important is what we find is that of late, and everybody knows that the immune system is an attractive target for cancer treatment, right? But what the challenges we are facing is in a lot of cases we are finding there is a immune suppression. There is the immune therapy is not, the tumors are not responsive to immune therapy. So what we are looking at here, the data shows that there is this group of certain things that are going on in the immune system in oral tumors, which might be important. And if we can work on these in future, probably something good might happen. Oral tumors are hot tumors as far as the scientific literature goes, but oral tumors have a very poor prognosis.
Hello. Am I audible? Please. Hi, sir. So it's a wonderful talk. So I have a very simple question that in one of your slides you have mentioned regarding the fetal, and in which you have calculated the trajectory of the cells, right? So can you put some light on what is the meaning of fetal there and how the trajectory was calculated?
What is the meaning of
Fetal cells.
fetal? So there are certain group of malignant cells which are expressing genes which are normally seen in fetal condition. So these genes are not expressed when somebody is crosses that fetal stage. So they are shut down. But what these malignant cells are doing is in these cells the fetal genes are getting switched on, and that probably might be related to immune evasion.
With respect to expression and analysis, beautiful work in profiling.
Please go to mic.
Can you hear me now?
Yeah.
Thing is, even the EMT has explained, there are population of cells. What I wish to know from you, comment on what is its role in genetic variation and transformation with respect to chemoresistance of developing therapeutic.
So EMT is notorious, right? It is associated with metastasis. That the EMT enables the malignant cells to go and lodge in other parts of the body and then seed new tumor development. What recently has been happening is people are understanding that there is the EMT is a whole process, and within, so that if you can catch using this method cells which are undergoing the transition, so you see partial EMT signatures, and those actually tell you that these things are going on there. So if you have metastasis, you will also have associated with poor drug response. So partial EMT signatures are something that is coming up. We are seeing that very much in this some of the cells, malignant cells are expressing, and we think that this can have again contribute to the poor prognosis of oral cancer. But this is in transition. The cells are in transition from not fully gone to the mesenchymal.
Arindam here. Very good work and a very interesting. Definitely lucky To the mic closer. Actually, you don't have any clinicians here because OSM and OSMF are actually they're still a long debate. And it's good that you have actually demonstrated that yes, we can actually cross it. But one of the issue is that what was the source of your sample? Was it a biopsy sample or?
These were biopsy samples.
Yeah. So in biopsy sample, you also have a lot of control samples, healthy samples. So it's better that if you can demonstrate that single cell data between the healthy data, healthy sample, and your cancer sample. Or one way is that you can do a separate of the cancer cell data by single cell.
So from our data, which I could talk about and which we are working on, is that when you look at the epithelial cells, we have epithelial cell markers. You can classify epithelial cells, but there is no such you can label malignant cells. So there are different ways of analyzing the data to classify malignant cells. We came up with our own method also, which we published last year in our first data set. So where we are looking at gene expression differences in an Euclidean distance Pachuda's brainchild. So we can very well demarcate malignant epithelial cells. So in this data set is sitting the whole lot of non-malignant epithelial cells. So coming to your question, that is where we are looking to understand how these malignant epithelial cells differ from the non-malignant epithelial cells, which generally we throw out of analysis.
And when you talk about the cell state, it could also become the epigenetic mechanism that cell state differs.
What I am looking at is a transcriptome, which is a readout of how the cells are behaving. So we can also look into single cell proteomics. We can also, epigenetics is of course controlling the expression, and in not here, but in another part of my research, we are actually using DNA methylation data and ataxic data, single cell ataxic data with the single cell RNA-seq. Not here.
So thank you very much, both of you, for the talk and for the question. Thanks again, Arindam, for nice talk. Now I hand it over to Dr. Kapadia for the next talk.
The next speaker is Dr. Gaurav Sharma, and he'll be talking about rating variation tests of multiple years. You have 20 minutes.